Okay, so what I want to do is go over this paper. It's not really a paper, it's more commentary that was written by this Carlos A. Perez, who's a medical doctor, and I don't know exactly where he works. He works at the Division of Multiple Sclerosis and Neuroimmunology, the Department of Neurology at the University of Texas Health Science Center at Houston. But before that, I'm going to talk about this drink that I made. So this is a watermelon drink. It's just take watermelon, take white wine, blend it together. You can add a little honey, a little lemon juice, maybe a little salt if you want, and yeah, turned out pretty well. It's the evening over here, so I can do this. So, why do I want to go over this paper? So, looking ahead, the risk of neurologic complications due to COVID-19. So, if you've watched any of the other videos that I put out earlier this day, you know that I currently have a very large concern of how these coronaviruses, explicitly the SARS-CoV-2, is able to basically infiltrate your central nervous system, or at least cause the production of autoantibodies that are going to target your neurons, or at least the proteins that are on the surfaces of your neurons, which could result in diseases such as multiple sclerosis, Alzheimer's, Parkinson's, basically the whole laundry list of neurodegenerative disorders. Guillain-Barré syndrome would be another example. So why am I concerned is that even though we can heal from these diseases, we are probably going to have to deal with an epidemic of neurological and other autoimmune complications years down the road as a result of us not caring about this SARS-CoV-2 pandemic. So if you can imagine, this SARS-CoV-2 is like the other coronaviruses, but on steroids as far as how infectious it is and how basically how able it is to infect cells and how able it is to basically destroy or ram through the immune system. So this is why I'm concerned, is that, for instance, with the SARS coronavirus, I think it's like OC43 or something like that, I forget the exact name of it, we're presuming with some genetic analyses, we've determined that it probably crossed over into the human population probably around in the late 1900s, so about 1890, which would correspond at least with one of the plagues that took place that killed about 1 million people in the world at the time. So my concern is this SARS-CoV-2 coronavirus is like that coronavirus, except far worse in its ability to kill more people and elicit a stronger immune response. So with that, sorry about this. With that, I want to go over this particular paper, looking ahead, the risk of neurological complications due to COVID-19. I think that we are probably going to see a lot of the similar, we're probably going to experience a lot of clinical complications or sequelae that happen after the fact that will result in a lot of pain and suffering for the entire world, and we are not even considering that right now, because we're only focused on the mortality rate, when really there's probably a lot more other conditions that need to be considered. So with that, I'm going to begin. So abstract: the rapid spread of SARS-CoV-2 caused by the the rapid spread of COVID-19 caused by SARS-CoV-2 has become a public health emergency of international concern. The outbreak was characterized as a pandemic by the World Health Organization in March 2020. That really should have been done in February, but I digress. The most characteristic symptom of patients with COVID-19 is respiratory distress. Some patients may also show neurologic signs and symptoms ranging from headache, nausea, vomiting, and confusion to anosmia, which is the inability to smell properly, ageusia, which is the inability to taste properly, encephalitis, and stroke. So coronaviruses are known pathogens with neuroinvasive potential. So neuroinvasive, the coronaviruses are known to be able to get into the nerves. So maybe they are not in detectable concentrations shortly after they get into the nerves, but they are known to be able to infiltrate the nerves. So, and that can also be problematic as far as your immune system is concerned, because For one, the central nervous system is an immunoprivileged site. So that means your immune system is not as able to attack those areas as easily as it can attack, say, normal body tissue. So that's one thing. Another problem is that if you do end up having inflammation of the neurons, you can actually start generating autoantibodies against neuronal proteins, and the generation of these autoantibodies can cause neurodegenerative diseases later in life. So that's another problem that we have to deal with. And then a third problem I can think of is that if these viruses are mimicking some of the proteins that are found within the brain as well, that could be another way at which you can develop autoimmunity against neurological neural proteins. So let's continue. So there's increasing evidence that coronavirus infections are not always confined to the respiratory tract. Central nervous system involvement can occur in susceptible individuals and may contribute overall morbidity and mortality in the acute setting. So additional post-infectious immune-mediated complications in the convalescent period. So after you've already healed, so after you have recovered from the coronavirus, there might be some immune-mediated complications which are possible. So awareness and recognition of neurologic manifestations is essential to guide therapeutic decision-making as the current outbreak continues to unfold. So perhaps there won't even be that very many neurological manifestations of SARS-CoV-2. However, there might be some. And we need to be fully aware as a society of what are the sequelae, or what are the sequels of complications that might arise from this disease. So we want to have informed consent, right? We want to be informed so that we can make our own decisions on what to do with our lives. We want to do the risk-benefit analysis of going out in public, the risk-benefit analysis of keeping the economy going for certain sectors or certain sectors of the economy. Let's do those risk-benefit analyses. But if we are not fully aware of all the risks, we might make the wrong decisions as far as what we should and should not do. Okay, so I know that there's a big discussion going around right now as far as should we open up schools? Should we allow children to congregate, right? So in the countries that have allowed that to happen, they really did, they cared about the community transmission, and as a result, it didn't matter as much to have the children still going into school. However, in the United States, they are allowing there to be significant community transmission, which means if they do end up opening the schools, that community transmission might also translate into excessive transmission within the schools as well. So one thing that we want to consider is if there are long-term neurological consequences to contracting SARS-CoV-2, we would want to use that in our risk-benefit analyses to determine whether or not that is a safe thing to do. So let's continue. So the virology and the taxonomy. And let us rank. That's pretty good. The watermelon that came from tasted really bad, but as a drink, it's decent, right? Anyway, SARS-CoV-2 belongs to the broad family of coronaviruses. It is an enveloped, which means that it has a cellular membrane or a phospholipid bilayer that is surrounding it. It is positive sense, which means the genetic material can directly be read by the ribosomes in order to produce a protein. So this virus is single-stranded RNA, which means this single-stranded RNA will be able to go in and then be read by the ribosomes of the cell, and then the reading of the ribosomes is going to cause the translation of the mRNA or of the RNA of the virus into fully functional proteins, which can then make up the replicase machinery, the machinery to duplicate the virus genetic material. It can create the spike protein. It can create the nucleocapsid protein, which is a protein that helps pack together the genetic material for the new developing viruses. So let's continue. And this has a diameter of about 60 to 140 nanometers. The viral envelope consists of a lipid bilayer with 4 structural proteins known as the S protein, the envelope protein, the membrane protein, and the nucleocapsid protein. Interactions between the spike protein and its host cell receptor is essential for virulence and infectivity. So the spike protein is a protein that is the one that's doing the binding in order to allow the virus to infect the cell. Anyways, similar to SARS, the causative agent of SARS global outbreak in 2003, and MERS, SARS-CoV-2 is a member of the genus Beta coronavirus. So genomic sequence analysis of SARS-CoV-2 has shown 82% sequence identity with SARS-CoV and an estimated mutation rate of approximately, how to phrase that, 1 out of 1,000 substitutions per site per year. So throughout every single year, we would presume that when you start with one strain of a coronavirus, and then as a year progresses of it infecting humans and continuing, we're going to expect about 1 out of every 1,000 nucleotides of the genetic material. And remember, the nucleotides are the letters of the genetic material. About 1 out of 1,000 nucleotides are going to be different in the entirety of the genome of the SARS-CoV-2 coronavirus from one year to the next. Granted, that's what it is now. That number theoretically could change. So these are all somewhat mutable numbers. Anyways, although mutation in the spike protein is the likely culprit for the recent introduction of SARS-CoV-2 into the human population, sequence variation among various isolates remains low, and a few mutation hotspots, so frequently mutations will occur in hotspots of the DNA that tend to be a little bit more, what would the word be? The changes that happen there are probably not as... The DNA, the genetic material itself, which is RNA, might be packed in a way that allows certain areas to be mutated more easily than others, and then also sometimes that RNA could also correspond to areas that are not as important to be conserved, and so therefore they have a higher mutation rate from generation to generation. Regardless, a few mutation hotspots have now been described. These advances should help facilitate vaccine design, clinical monitoring, and management of COVID-19. So if we, for say, do vaccine design, we would do it with one of the mutation cold spots. We would do it on an area that is not mutating rapidly, because then the antibodies that are generated for that strain are going to be more likely to be protective against a new strain. Anyways, the neuroinvasive nature of human coronaviruses has been previously described, and neurotoxicity may occur as a result of indirect, direct, and post-infectious complications. So they could be direct, where the virus is infecting the neurons in of themselves, or they could be indirect, in that the virus is causing something that ends up screwing something up in the brain from a completely unrelated, from a completely different effect that is not from the virus infecting the neurons themselves. And then there could be post-infectious complications, that the virus is causing some sort of physiological change in the body, and that physiological change ends up screwing up the neurons in some way as well. So here are the indirect neurological complications. So coronaviruses generally cause respiratory and enteric, which means of the intestines, respiratory and enteric diseases in animals and humans. In a report of 44,500 confirmed COVID-19 patients by the Chinese CDC and prevention, 81% of cases consisted of mild pneumonia. 14% were characterized by more severe disease, including dyspnea and hypoxia. So dyspnea is the inability to breathe properly, and hypoxia is having too low of oxygen in the blood, and or not getting enough oxygen. And 5% had respiratory failure, shock, and multi-organ dysfunction. So sepsis, heart failure, and septic shock are among the most frequently reported systemic complications of COVID-19, and extrapulmonary or outside of the lung involvement of SARS-CoV-2 may induce neurological signs and symptoms. So a recent report of 99 COVID-19 patients in Wuhan, China, describe the presence of neurologic manifestations, including confusion, headache, confusion and headache. In a different report of 214 COVID-19 patients, 36.4% were found to have neurologic manifestations at presentation, including headache, nausea, vomiting, impaired consciousness, ataxia, which is the inability to move properly, acute cerebrovascular disease, which would be a disease of the blood vessels that are in the brain, I believe, but let me look this up so that I'm not spouting nonsense. The internet is not the best right now. So cerebrovascular disease refers to a group of conditions or disorders that affect the blood vessels and the blood supply to the brain. Okay, so that is true. Okay, sorry, I'm running like 10 programs at once right now, so my ability to switch between tabs is compromised. Anyways, secondary neurologic manifestations of COVID-19 are thought to result from the widespread dysregulation of homeostasis due to pulmonary of the lungs, renal of the kidneys, hepatic of the liver, and cardiovascular injury. So, additionally, the direct and indirect cardiotoxicity, toxicity of the heart, secondary to excessive systemic pro-inflammatory stimulation. This is also known as the cytokine storm. So the cytokines are cellular signals, protein cellular signals that cells will release just to talk with each other. And there are a lot of pro-inflammatory cytokines, which they can basically be positive feedback loops and cause the creation of more pro-inflammatory cytokines. Regardless, you can have excessive systemic pro-inflammatory stimulation from these cytokines all over the body, causing more and more inflammation. That could be something that can cause direct and indirect cardiotoxicity. There's also the problem with hypercoagulability, so these blood clots that are forming all the time, and there's also direct myocardial invasion, and these are associated with myocardial infarction, heart failure, and arrhythmias, which are important risk factors for stroke. So they're saying here that there are a lot of these systemic problems that could be screwing up the heart, and by screwing up the heart, you could also be screwing up the brain as well. So here are the direct neurological complications. So in previous SARS-CoV, this is the original SARS infections, the post-mortem or after-death studies have reported the presence of viral RNA within the brain tissue of affected individuals. So the acutely ill patients with SARS-CoV have also demonstrated the presence of the virus in cerebrospinal fluid, also known as the CSF. So the experimental animal studies using transgenic mice have further revealed that following intranasal administration of SARS-CoV, the virus can enter the brain. So if you give these mice the SARS, the original SARS coronavirus, and put it into their nose, the virus will have the ability to enter the brain. And so this is in accordance with some of the other coronaviruses that we know. These other coronaviruses are also able to get into the cerebrospinal fluid. They're also able to get into the central nervous system in some way. And one of the methods by which this can happen is through the olfactory bulb, or the nerves that are in this cavity of your face. There are some nerves there, and then these coronaviruses can possibly infect those nerves and then actually transport through the nerves, through the axons, by diffusion or by active transport. They can be transported deeper into the brain. So this original SARS is doing similar things that we know that other human coronaviruses can do, and SARS-CoV-2 is probably going to be doing the exact same thing as well. So the virus, and therefore it may spread to specific areas of the central nervous system, including the thalamus and the brainstem. So in addition, cases of HCoV-related meningitis, encephalitis, and acute flaccid paralysis have been reported over the years. So these are neurological conditions that have been reported of the original SARS through direct infiltration into the nervous system. So similar to SARS-CoV. Oh, and I also need to mention as well is that your brain is an immune privileged site. And what do I mean by that? I mean that the immune system does not function as easily or as readily in the nervous system as it does in other areas. So even if you have antibodies, this is what I need to stress. Even if you have immunity, if you have antibodies and if you have this adaptive immune response against SARS-CoV-2, and maybe it is effective, that doesn't necessarily mean that if this virus is able to infect you again, how do I phrase that? So when you get reinfected, you hopefully, if your immune system functions ideally, you don't get sick because your immune system is able to search and destroy the virus the second time it has infected you, and you do not show any pathology. Okay? Now, you run the risk of this virus if you breathe it into your nose, or if it can get into areas in which it can infect the nerves. It is possible, it is theoretically possible that these viruses might still be able to get into the brain before your adaptive immune response is able to search and destroy it. So that is possible. Maybe the immune system does get activated, and maybe it does end up attacking the virus particles that are on one side of the blood-brain barrier, but it is still possible that these viruses might be able to, through diffusion and through axonal transport, get into the central nervous system, where it is going to be much more difficult for your immune system to fight it. The immune system is suppressed in the brain, so be careful about viruses that are able to get in there. I digress. So similar to SARS-CoV, SARS-CoV-2 appears to enter the human host by binding to the angiotensin-converting enzyme 2 receptor on the surface of epithelial cells in the lung, intestine, kidney, and blood vessels. Granted, ACE2 is all over the body, so it's not just there. The exact mechanism by which SARS-CoV enters the central nervous system remains unclear, but it may involve disruption of the nasal epithelium and entry through the olfactory bulb with subsequent retrograde transsynaptic spread. So transsynaptic means going over the synapses of the neuron. So basically, the virus is basically just going through the nerves, and then it gets to an axon, which is the junction between nerves, and then it finds a way to jump from one nerve to another. And through that mechanism, they might be able to infiltrate into the central nervous system. So be wary about that. Interestingly, evidence of neuronal expression of ACE2 receptor has recently emerged from the human protein database. So I believe a lot of research went into finding out where the ACE2 receptor is expressed, and so they recently said, oh yes, the ACE2 receptor is expressed in neurons as well. So the neurons are theoretically vulnerable to ACE2, or they're vulnerable to SARS-CoV-2 and presumably also SARS. But a clear role for this receptor and HCoV entry into the central nervous system has not been established. A recent report describing the ability of HCoV to induce direct neuronal injury with brainstem cardiorespiratory centers in experimental animal models has raised concern that SARS-CoV-2 may be partially responsible for the acute respiratory failure reported in patients with COVID-19. So in addition, an association between the COVID-19 and early onset olfactory and gustatory dysfunction, and I think gustatory means of the digestive tract, but let me, the sense of taste. Okay, so basically they screw up your sense of smell and your sense of taste. The dysfunction of the olfactory and the gustatory has been reported recently. So the American Academy of Otolaryngology, sorry about that. This proposed that anosmia, hyposmia, and dysgeusia should be added to the list of COVID-19 screening symptoms and urged precautionary isolation for individuals with these symptoms, even in the absence of respiratory disease. And anosmia, can't smell, hyposmia, the lowered sense of smell, and dysgeusia, the inability to taste correctly. And although changes in smell and taste perception are known complications of a range of viral infections, these observations raise the question of whether these changes arise from secondary inflammatory responses or are due to the SARS-CoV-2 related dysfunction in olfactory and gustatory processing mechanisms. So there is the possibility that there might be a secondary inflammatory response that is causing these sensory complications, or it might also be the result of the virus itself infecting these areas. So I would presume it's, since even the mild cases, there seems to be the lack of taste and the lack of smell. So in the mild cases, that would at least scream to me that it was probably the virus itself before the inflammation, but who knows exactly what's going on. So of note, the ACE2 is also expressed in microvillar and Bowman gland cells. Microvillar, the microvilla? So let me look at the Wikipedia. The microvillus. Brush border cells. Okay, sorry about this. I think, if I remember correctly, this is the... So microvilla are covered in the plasma membrane, which encloses cytoplasm and microfilaments. So though these are extracellular extensions, there are little or no cellular organelles present in the microvilli. Thousands of microvilli form a structure called the brush border that is found in the apical surfaces of some epithelial cells. So, yeah, I'm surprised that they're calling it the microvillar cells, because I thought that the microvilla were not a cell in themselves. I thought they were protrusions from the cells, which I think. So let's look up Bowman's gland cells. So these are the olfactory cells, also known as the Bowman's gland cells. So these are they're in the olfactory mucosa beneath the olfactory epithelium in the, so these are a type of smelling cell. So ACE2 is located there, so that's probably not beneficial for, that would indicate that those are sensitive cells at least. So smelling might be something that could get you infected. The central nervous system entry has not yet been established. So a genetic predisposition for an increased risk of COVID-19 related neurological complications, which may be partially due to ACE2 polymorphisms linked to these conditions in previous studies, should be further investigated. So the post-infection neurologic complications. So following the acute phase of the infection, the presence and persistence of human coronaviruses within the central nervous system may lead to misdirect host immune responses. So one thing that I'd like to mention, that another paper that I talked about earlier today was that in multiple sclerosis patients, when they took the cerebrospinal fluid samples from them and did PCR on them, they were able to find in most multiple sclerosis patients, they were able to find at least one of the two human coronaviruses, the common coronaviruses that are known to circulate throughout the population year after year after year. Granted, to find it in the central nervous system or the cerebrospinal fluid would indicate that either they had an ongoing infection and it went into the cerebrospinal fluid somehow, or it is existing in the cerebrospinal fluid for a long period of time, even after they've recovered from that coronavirus. So, so here they're talking about the presence and the persistence of human coronaviruses within the central nervous system. This may lead to misdirected host immune responses. So if the virus is just existing there for a long period of time, this might cause misdirection of the immune system. So this, which could be associated with autoimmune, inflammatory, and demyelinating syndromes in susceptible individuals. And this is not just in the human coronaviruses. So a lot of people do not know that coronaviruses in a lot of our animal models are frequently used to induce encephalitis. They're used to induce inflammation of the brain. They're used to induce multiple sclerosis. They're used to induce these demyelinating events. Very, very frequently, we use coronaviruses in our animal models to cause these autoimmune diseases. And by causing these autoimmune diseases, we're like, oh, yes, we can use these as a model to study these diseases in humans. But this is just screaming to me that if we are using these coronaviruses to cause these problems in animals so that we can study these diseases in the animals, then we are now the experiment. Now that we have this pandemic going on, we are now the experiment to see whether or not we are going to get all these autoimmune diseases from this new coronavirus. So granted, with the previous coronaviruses, those have already, when they crossed over into humans, they have accomplished their play, right? So when these first jump from animals to humans, they are going to sweep through the population and kill all the susceptible people, and then therefore we undergo successive rounds of evolution by having mass death. So with the most recent human coronavirus that has crossed over into humans around the late 1800s, it is presumed that there is a plague that was associated with that killed about 1 million people. And 1 million people at that time was a much larger percentage of the population as it is now. So that happened to humankind before. However, we have gone through the successive killing, the culling, that has allowed us to evolve to live more in harmony with that particular coronavirus. And I think that's the OC43 strain, which is a relative of the cow or the cattle coronavirus. Bovine is the word they use. So knowing that this is something that coronaviruses they explicitly do this, and we study these coronaviruses to cause these autoimmune diseases in other animals so that we can study the pathology of those other animals and try to translate it back into humans. Well, crap. Now we are the experiment. We are the experiment with a new coronavirus, and I am not excited to see the results of it. Okay? So, sorry, another tangent. I'm a little frustrated that this is happening and people just do not care or do not even know that these coronaviruses are causing these autoimmune diseases. So, continuing. So, in support of this hypothesis, and also, let me go back a sentence. So following the acute phase of the infection, the presence and persistence of the human coronaviruses, which, these coronaviruses take so long for your immune system to get rid of them. They persist, especially in the central nervous system. The persistence of human coronaviruses within the central nervous system may lead to misdirected host immune responses, which could be associated with autoimmune, inflammatory, and demyelinating syndrome. So demyelinating means that it's removing the myelin sheath, and the myelin sheath is the protein that is surrounding the axons of the neurons. And so this myelin sheath helps protect the electrical impulses. It allows the nerves to have proper electrical impulses that go through those axons, right? And so this is how you think. This is how you rationalize. This is how you survive, really. When you remove those myelin sheaths in a demyelinating syndrome, then the electrical signals cannot pass through that axon as easily, and then you end up with some form of pathology, right? So people who take methamphetamines and are addicted to methamphetamines, a lot of their the myelin sheaths on their nerve cells are screwed up, and as a result, they cannot send signals to and from one nerve to another as quickly as they can in a healthy individual. So continuing, and we use these coronaviruses to cause demyelinating events in animal models, and we have a lot of presumption that the normal human coronaviruses are causing these demyelinating syndromes in us as well, however in low amounts, because we have theoretically evolved or co-evolved after successive rounds of evolution to basically live in harmony with them a little bit more. However, harmony meaning that you get a neurological condition late in life, I don't think that is true harmony, but it's harmonious enough for, since we don't have children late into life, it is harmonious enough for us to still multiply with it, regardless. So in support of this hypothesis, the presence of human coronavirus RNA in the cerebrospinal fluid of patients with acute disseminated encephalomyelitis, this is called ADEM, and multiple sclerosis, this has been reported. So finding the human coronavirus RNA has been shown in multiple encephalomyelitis. This would be inflammation of the brain, and acute inflammation in the brain, and then also with multiple sclerosis as well. So human coronaviruses in the fluid has been linked and implicated in causing these diseases in humans. So HCOV infections have also been associated with other post-infectious syndromes, post-infectious long after the infection. So this is including encephalitis, inflammation of the brain, Guillain-Barré syndrome, which is numbness. So this is a demyelinating event of the peripheral nervous tissue, which ends up having numb areas, causing numb areas of the body, and a plethora of other diseases. And this is also implicated to be a an autoimmune disease as well. Anyways, proposed pathogenic mechanisms include the molecular mimicry. Keep this in mind, molecular mimicry. These coronaviruses might be mimicking the shape and structure of some of the proteins that are within your body. So that it's mimicking you. So if you make an antibody against a mimic, that antibody is not just going to attack that virus, it is also going to attack your own body, okay? So this is something that we are greatly concerned about. So the proposed pathogenic mechanisms include the molecular mimicry between HCOVs and the myelin basic protein. And coronaviruses are not just implicated in mimicking this myelin basic protein. They seem to be mimicking a lot of proteins in the body, not just the myelin basic protein. So there's also direct invasion of leukocytes and other immune cells. Notably, the dysregulation of the ACE2 receptor has also been implicated in the development of the experimental autoimmune encephalomyelitis. So EAE, so here a murine disease model that resembles multiple sclerosis. So experimental, I think it's also sometimes called experimentally acquired autoimmune encephalomyelitis. So basically this is when we give a coronavirus to a mouse, and then this coronavirus ends up causing an autoimmune response where those autoimmune antibodies end up destroying the myelin sheaths of the nerve cells of the mouse, and then causes, unfortunately, encephalomyelitis. So this really bad acute inflammation of the brain, which can cause paralysis, it can cause suffering on the creature, and so we do not want to see this happen to humans as well. So I need to also make mention that we use mice models. So since mice live for such a short period of time, we do a lot of experiments on the mice, hoping that since the mouse goes through its life cycle so much faster, if we do something to the mouse, we are hopefully going to see the effects of that experiment on the mouse within a very short period of time, because the mice do not live that long. So in humans, since we live so much longer, if we see an effect on a mouse in a short period of time, we would expect to see that happening in humans after a much longer period of time. So we are worried about, I am worried, that this experimental autoimmune encephalomyelitis that we see relatively quickly giving mice these coronaviruses, for us to fully experience the equivalent of that, which would be multiple sclerosis, that would take a longer period of time for that pathology to come to fruition, right? So we won't even know. We won't even know until years down the road if this is actually going to happen, which means it's better not to deal with it in the first place. Sorry. Let me continue. So, immunopathogenesis. So, a structural and phylogenetic similarities between the SARS-CoV-2 and SARS-CoV suggest that the known pathophysiologic mechanisms for related human coronaviruses are likely applicable to SARS-CoV-2. So following invasion of the lung epithelium, SARS-CoV directly infects T cells and macrophages. So your T cells and your macrophages are susceptible to the original SARS. And this is contributing to lymphopenia, so low levels of the lymphocytes, with reduced CD4 positive and CD8 cell counts. And CD4 positive and CD8 positive, these are just different differentiated T cells. And counts and dysregulation of the normal adaptive immune responses. So you basically have a screwed up adaptive immune response as a result of the original SARS infecting you, which granted was far deadlier, right? So the original SARS, about 1 out of 3 people or 1 out of 5 people ended up dying. I don't remember the exact number. Anyways, excessive production of pro-inflammatory cytokines, including interleukins 1 beta, interleukin 6, interleukin 10, and tumor necrosis factor alpha. Don't worry too much about those. Basically, there was excessive production of the pro-inflammatory cytokines, with subsequent downregulation of anti-inflammatory cytokines such as interferon gamma. This is thought to contribute to the excessive inflammation and activation of the pro-apoptotic pathway. So apoptosis is when the cell is programmed to kill itself. So basically, if a cell gets enough signal to kill itself, it will then undergo programmed cell death, also known as apoptosis. And if you have way too many or a dysregulated pro-inflammatory response, you can get a lot of apoptosis. You can get dysregulated apoptosis as well. So in some cases, the adaptive immune responses might become directed against host epitopes. So host epitopes, these are an epitope is the area of a protein or an antigen that a particular antibody will bind to. So think of epitopology. So this is the topology of the antigen that is on the surface, the surface of the antigen, the topology of it that these antibodies will bind to. So this results in the post-infectious autoimmune reactions that prolong the inflammatory phase and induce tissue destruction even after the virus has been cleared. So remember, if you go into a hyper-inflammatory response, you end up, without getting too technical, you have immune cells in your body that are always taking up the antigens in its environment, including your human antigens or bacterial antigens. They're just taking them up, the protein ones. And they will take these antigens and they will present them on the surface. And so when you are happy, when you are not inflamed, those antigens will be associated with the normal state in your body. It will be an anti-inflammatory antigen. So as the immune cells bind to those antigens on the antigen-presenting cells, they realize, oh, these are safe antigens, therefore I do not need to attack them. And then they, so yeah, that is with the dendrites, if I'm remembering correctly. But if you get into a very, very pro-inflammatory state, then what happens is these dendrites, these antigen-presenting cells, will end up maturing, and they go from their safe form into their reactionary form. They become reactionary types of immune cells or antigen-presenting cells. And then they basically convince, something happened, but they basically convince the immune cell to attack whatever antigens are being presented. And if some of those antigens are from your own body, then you are actually training your immune system to attack your own body. And so that could be a prolonged inflammatory phase that is post-infectious, that comes long after the virus has already been cleared from the body. So I digress. So data from the experimental animal study suggests that antibodies might also contribute to immunopathogenesis, as increased uptake of coronavirus by macrophages can lead to macrophage activation and secretion of cytokines and other chemokines. So if you get more of these antibodies that are being secreted into the environment, you might actually become even more pro-inflamed as well. So in addition, coronavirus-specific T cells and antibodies have been shown to activate macrophages, resulting in their migration into the central nervous system and ultimately in demyelination. And that is what we do not want. We do not want. So remember how I said the brain is an immune-privileged site? that means that it's difficult for the immune system to get into the brain, and that's because the brain is so sensitive. The brain does not want the immune system there whenever possible because the immune system is very damaging. So when the immune system gets kicked into overdrive, even the brain is not safe from the immune system, and then those immune cells can actually penetrate into the central nervous system, where they might actually result in more of these autoimmune diseases. So immunotherapy. So the assumption that the immunocompromised patients could be at increased risk for severe disease and mortality from COVID-19, presumably due to the lack of robust immune response. This has been a matter of recent debate. A recent report by Liang and colleagues suggested that the cancer patients might be at increased risk of infection and have a poorer prognosis than individuals without cancer. Conversely, it has also been suggested that the immunosuppressive states may actually help. They might actually help blunt the hyperinflammatory responses in patients with COVID-19. So I've done other videos where there were some case studies of people who were immunocompromised, where they had, I don't remember if it was toxumilabab, or they were on some drug, or they were given a drug previously which kills a lot of the immune cells in the body. It ends up putting you into an immunocompromised state in order to heal you from something, right? So there was a few case studies that were done of people who were immunocompromised but still had a very favorable outcome from contracting SARS-CoV-2. So this is indicating to me and to a lot of other people that perhaps specific types of immune compromised states might actually be beneficial for fighting off, or at least not damaging, what would the word be? It might not be detrimental to have certain areas of the immune system downregulated if you're fighting SARS-CoV-2. So, oh, I was right. So a multi-center randomized controlled trial of tocilizumab, which is an interleukin-6 receptor blocker, which is approved for the treatment of cytokine release syndrome. This has been approved in China for patients with COVID-19 pneumonia and elevated interleukin-6. And remember, interleukin-6 was one of the pro-inflammatory cytokines. However, these assumptions are largely based on available information from small cohorts, and current evidence remains insufficient to explain an association between immunosuppressed states and the risk for COVID-19. Nevertheless, these observations raise important questions regarding the safety and immune modulating therapies in patients with multiple sclerosis, myasthenia gravis. I don't know what that is. Let me look that up real quick so that I'm not. Nope. I do not want to print. Sorry about this. So myasthenia gravis, search Google. It's a neuromuscular disease that leads to varying degrees of skeletal muscle weakness. The most commonly affected muscles are those of the eye space and swallowing, so that's, yeah, not good. And other neuroimmune disorders. So in conjunction with the National Medical Advisory Committee and the National Multiple Sclerosis Society, they recently published recommendations regarding the use, continuation, and initiation of disease-modifying therapies, also known as DMTs, not to be confused with dimethyltryptamine, which is a hoot, I hear. Anyways, the Association of the British Neurologists, or ABN, they have also published general advice related to DMT, the disease-modifying therapies used in multiple sclerosis patients, and immunosuppressive drugs and other neuroimmunologic conditions. However, these recommendations are largely based on medical expertise, and more organized data is currently needed to understand the true risk of these disease-modifying therapies in multiple sclerosis patients, keeping in mind that immunosuppression in this population presents distinct challenges that differ from other immunocompromised subtypes. This is including HIV and cancer. So remember, these multiple sclerosis patients and other types of neuroimmunologic conditions, they have one type, or I should say they have a specific type of being immunocompromised. They're attacking a specific part of the immune system, whereas cancer and HIV patients, though they actually have a more encompassing destruction of their immune system that is not the same as these MS patients. So even though they're both immunocompromised, the immune system is very complicated, and so just because you compromise one part of the immune system doesn't mean that the other parts are not sufficient in of themselves to fight a specific disease. These cohorts might not be applicable to multiple sclerosis and other neurological conditions. So in light of the current lack of specific treatment, the USDA recently approved the use of COVID-19 convalescent plasma, which is plasma from the people who have already healed from COVID-19. They've used this in patients with severe life-threatening diseases requiring mechanical ventilation or continuous positive airway pressure, or CPAP, which has shown promising results in small preliminary uncontrolled case series. The use of this treatment modality is largely based on the premise that antibodies may be helpful in improving immune response against SARS-CoV-2. This might not be a perfect assumption to make. Just so that we are aware, sometimes cells can viruses can sometimes utilize antibodies to infiltrate different cells or infiltrate the immune cells. So antibodies are not always good. Keep this in mind. Antibodies are not always good. They can sometimes make disease worse. And one of the mechanisms is called antibody-dependent enhancement, also known as ADE, and a lot of other coronaviruses are implicated in being able to utilize this strategy in order to make the disease worse. So do not allow yourself to contract SARS-CoV-2 multiple times, okay? Just be aware of this. But the potential risk of such therapies remains unknown. So novel therapies to combat COVID-19 pandemic include vaccines. These should be designed to minimize pulmonary and systemic disease while optimizing an equilibrated anti-SARS-CoV-2 immune response, and dysregulation of normal host immune responses could also lead to untoward consequences. So, conclusion, taken together, neurotropism and neuropathogenic potential appear to be common features of HCOVs, human coronaviruses. As the full clinical spectrum of COVID-19 continues to be described, it is quite likely that SARS-CoV-2 possesses a similar potential for extrapulmonary or outside of the lung pathology and central nervous system invasion. I think this is pretty much established now. Like, we're pretty sure that the central nervous system can get screwed up by SARS-CoV-2 by now. So anyways, the putative role of ACE2 in the pathophysiology of COVID-19 has recently fueled concerns regarding the use of non-steroidal anti-inflammatory drugs, also known as NSAIDs, such as ibuprofen, which are known to induce upregulation of ACE2, and in theory may facilitate infection with COVID-19. Keep in mind, though, that on the other side is that these coronaviruses can actually remove ACE2 from the body, and that can actually cause a different type of a negative effect. So we aren't exactly sure whether or not we should add these NSAIDs to upregulate ACE2 in order to, what would the word be, restore the function of ACE2, because it appears as though the virus itself is able to get rid of some of them. Anyways, it would be reasonable to speculate that treatment with these ACE2 stimulating drugs could similarly increase the risk for severe and fatal COVID-19 in the central nervous system complications. So owing to the paucity or lack of current medical literature describing the neuropathogenic potential of human coronaviruses, the number of COVID-19 related neurological complications is likely underestimated. So we don't even know what the other human coronaviruses can do to the central nervous system. So since we have a lack of that information, we are going to be, we're at a disadvantage trying to figure out what COVID-19 can do to the nervous system. Anyways, a cross-disciplinary perspective is essential in preparing for the potential consequences of the current pandemic to human health, and awareness of the risk for acute and post-infectious neurologic complications and their contribution to COVID-19 related morbidity and mortality can help guide therapeutic decision-making and allow for delivery of individualized treatment as the number of reported cases continues to rise. So with that, I'm going to head on out. I'm starving, and I'm surprised that I didn't drink more of this, but it is what it is. So I like this. I wish that it went into a little bit more detail as far as how the immune system ends up attacking your own neurons, but granted, this is good for this is a good introduction, at least, for a lot of people. So yeah, I don't think I have that much to say, except be prepared for an uptick in neuropathologies. Just be prepared for an uptick in that. So, for one, I know that Elon Musk might be working on he's working on Neuralink, so that might actually be a good company to invest in right now, because if we end up having a lot of these Guillain-Barré syndrome or these pathological conditions that prevent us from being able to use our nerves properly, perhaps a company like Neuralink might show a lot of promise. Just be aware, though, that we might actually have to deal with a huge increase in, say, multiple sclerosis and Alzheimer's and other neuropathological diseases, just because we've allowed a new coronavirus to enter the fray. So it's not like we just got a new coronavirus. It's you have to deal with all the other coronaviruses, and then we got this fifth one that is now circulating throughout the entire population over and over again. So with that, I'm going to head on out. Thanks for listening, and hopefully I will see you tomorrow.